properly fixed samples to allow the target probe to enter the cell to hybridize to the target sequence. The specific signal then get picked up by the double Z probe pair will then be amplified by a series of amplification cascades, and the signal can then be visualized under the microscope as single punctate dots. Each dot represents a single copy of target RNA. The signal can then be quantified on a single cell level by manual counting or automated image analysis using image analysis software. Then let's talk about the key aspect of the RNAscope technology, which is the double Z probe design. There are three components of the Z. The lower region of the Z is an 18 to 25 base sequence that is complementary to your target RNA. The upper region is a 14 base pair tail sequence, a spacer sequence in between that links the two components of the probe. These are used in pairs. So in pairs, the upper region of the Z will provide 28 bases, which is the preamplifier binding sites, and is where the amplification cascades occurs. Both Z bind adjacent to each other to your target from the lower region. Here, move on to the right-hand side here. The yellow line represents your RNA. So if your RNA is intact under ideal pretreatment condition, the assay will hybridize up to 20 ZZ probe pair to your RNA. But for most samples, such as cancer FFPE samples, your RNA could be degraded and fragmented. The simplicity of this design, relatively short probe binding to the target, allow for binding to even shorter pieces of RNA. So our scientists have demonstrated that we only need 3 ZZ base pair to bind to the target in order to detect the signal. With the unique ZZ probe design, mRNA transcript detection of RNAscope assay is based on highly specific and robust signal amplification. To design a specific probe for your target of interest, we request to provide either a session number information or the exact sequence of your target. You could even indicate your region of interest for targeting with start and stop positions. Our bioinformatics scientists channel this information through the proprietary ACD alignment algorithm to select optimum sequence to ensure target specificity and uniformity of oligo hybridization in the assay. Our algorithm evaluates the melting temperature of each oligo in the pool to ensure no cross-hybridization with non-target RNA under stringent RNAscope conditions. With the standard probe design, the target probe set is a pool of 20 ZZ oligo pairs, and it binds to a 1 kb RNA target region. So now, how does the signal amplification work? So here you have your target transcript. When the probes are added, double ZZ probe pair land adjacent to each other. The amplifier will then bind to the upper region of the probe. Each preamplifier will then serially have 20 amplifiers bind to it. On each amplifier, there are 20 labeled probes. So we see 20 ZZ probe pair with 20 amplifiers and 20 labels. That is 8,000 label molecules per per 1 kb target transcript. In other words, the specific signal gets amplified 8,000-fold. So what about non-specific binding? If a single Z binds to non-specific RNA, the preamplifier cannot be stable on just 14 bases of the upper region. So the non-specific binding that may occur does not get amplified. This is how double ZZ design prevents background noise and contributes to specificity. So in theory, it's highly unlikely for two independent probes to hybridize to a non-specific target adjacent to each other. However, to further validate this probe design strategy, a set of probes targeting 18S ribosomal RNA was split into two groups, one with the left tail only, one with the right tail only. These two groups were used alone. The lack of signal when only one set was used or when the sense probe is used conformed the probe specificity. Another experiment up here showed using the sequential amplification system versus just a single preamplifier significantly increased the specific signal intensity. With regards to RNAscope publication and literature references, the Wang paper published in 2012 in the Journal of Molecular Diagnosis is the original paper that describes the RNAscope technology. Since 2011, our technology has been rapidly adopted by a wide variety of research applications. We have over 460 publications until May 2016. However, up to today, this number has increased to over 500. Just to name a few, the top research area includes cancer, infectious disease, and neuroscience. Now let's move on to our next section, focusing on how this technology can be implemented on automated platform. The RNAscope 2.5 VS assay is designed for use with the Discovery Ultra and the Discovery XT automated slide staining system by VENTANA Medical Systems. This assay is based on ACD patented signal amplification and background suppression technology just described in the previous section. So with the VENTANA automated system, you essentially get to apply the same chemistry and get the same results with much less hands-on time. As compared to manual assay, automated assays significantly save hands-on time from 8 hours for manual assay down to only 2 hours for a semi-automated and 30 minutes for a fully automated assay. Meanwhile, automated assay can achieve the same sensitivity and specificity. Running the automated assay has two main components. One is the instrument, the other is the reagent. So for instrument, the compatible instruments are VENTANA Discovery Ultra and the VENTANA Discovery XT. With this instrument, you are also provided with an option of the fully automated or the semi-automated workflow, which means permeabilization step can be done either online or offline. Next, let's review the software requirements that you will need to have in order to run the RNAscope automated assay. mRNA DAB Discovery XT or mRNA Red Discovery XT software is required to run either the brown or the red assay on Discovery XT. For Ultra, mRNA Discovery Ultra DAB or Red 4.0 software is required. Now that we are aware of the instrument and the software requirements, let's review the reagents and material components for running the automated assay. Automating RNAscope assay is as easy as step 1 to step 4 listed here. So you will need to order the reagents and materials partially from ACD that's listed for step 1 to 3 and partially from VENTANA Medical Systems, that's the step 4. We will discuss this in more details in the next couple of slides. So this slide summarizes all the reagents you will need to order from ACD to run automated assay on VENTANA. First is the reagent kit. So each reagent kit consists of these 3 components. Each reagent kit is enough to run 60 slides. Second, you will also need the species-specific positive control and negative control probe, as well as the target probe for your gene of interest. Each probe is enough to stain 30 slides. Please refer to the table on the right here for more detailed information, including storage, temperature, and cut log numbers. This slide summarizes all the reagents and materials you will need to order from VENTANA Medical Systems. First is the detection kit, which is provided to you pre-filled, which is highlighted in blue here. And the empty dispensers highlighted in orange here are used to add the reagents that you receive in the ACD reagent kit. Please refer to the bottom information here for all the cut-log information. Finally, let's review the software nomenclature briefly before our next section of setting up the assay. May I have your attention on the first and the second column of this table here. So for target retrieval reagents, it's called cell conditioning in VENTANA software, and it's used for offline boiling in the semi-automated workflow. RNAscope 2.5 VS target retrieval is also called cell conditioning in the software, but the difference here is it's used for online pretreatment that is part of the fully automated workflow. RNAscope 2.5 VS mRNA PRETREAT 3 protease is used as PRETREAT 3 in VENTANA software. It's required for both offline and online pretreatment steps. Finally, the RNAscope VS accessory kit is used for counterstain 1 and counterstain 2 steps in the VENTANA software. Let's start with the semi-automated workflow. I will be providing an overview of the process with only a few key steps discussed in detail. This workflow involves baking, deparaffinization, and partial sample pretreatment offline. So the semi-automated RNAscope VS assay workflow consists of 4 main parts. In the pre-run preparation part, it includes preparing the properly fixed samples, preparing instrument reagents, making sure bulk reagents are filled, and setting up the protocol as well as printing the barcode. Please note that the recommended fixation condition for FFPE samples is 10% MBF for 24 hours at room temperature. The offline sample pretreatment step includes offline baking and deparaffinization, as well as the offline target retrieval boiling step, followed by probe hybridization, signal amplification and detection on the instrument, and finally finish with dehydration and mounting the slide. This step is all done offline as well. While setting up the protocol on Discovery XT, please refer to the user manual for recommended conditions. But here, just to highlight a few key points here. First, please make sure the mRNA DAB Discovery XT software is selected if you are running brown assay. Second, it's very important to uncheck the baking, deparaffinization, and cell conditioning steps, since these steps have been done offline and no need to repeat these steps on the instrument. And next, the standard pretreatment condition is 30 degrees for 16 minutes. We always recommend starting with standard pretreatment condition and optimize if necessary. Finally, the standard AMP5 incubation time is 32 minutes, and the AMP5 incubation time can be adjusted to improve detection results in some cases. Furthermore, please make sure the probe hybridization temperature is at 43 degrees, and the incubation time for AMP5 step, again, is at 32 minutes. And also, please make sure the counter stain time are all set at 4 minutes. So moving on to VENTANA Ultra, same again, please remember to keep these 3 steps unchecked so that you are not doubling the pretreatment. And then please follow the same post-processing guidelines as discussed earlier. Once the protocol is set up, next please proceed to preparing your samples. Let's review the recommended offline boiling step. Here's a typical hot plate setup for doing the target retrieval step offline. We recommend closely monitoring the boiling temperature and time during the entire boiling step. Please maintain the temperature at around 100 degrees and monitor the temperature with a thermometer. Once the time is up, please immediately remove the slides and put them in room temperature DI water. It's very important here, please do not cool the slides in the boiling target retrieval buffer. Next, let's review the offline boiling guidance. So for any given tissue, please use the same condition with target retrieval buffer that worked for PRETREAT 2 with 2.0 RNAscope chemistry. If the tissue is new to you, please refer to the table in the user manual as initial guidance. So this table is also shown on the right here. The conditions have been worked out by our R&D scientists. For protease time, we always recommend start with 16 minutes, and since we find this time point is optimum for most samples, so this time can be adjusted if needed. So before you start to run, we highly recommend checking the following 4 points to ensure successful results. So first, please ensure heater pads are completely dry. Second, fill and prime dispensers with the correct reagent. And third, please check the bulk reagent level and making sure and the waste containers are empty. So it's very important, the last point is before leaving the instrument unattended, please ensure all reagents and slides are successfully registered. So one tip we would like to draw your attention here is to take the labels off the ACD reagent container and stick it on the dispenser. So this way will ensure no mix-up of reagents and also maintain the kit lot-to-lot consistency. So after the run is complete on the instrument, first thing, please wash the slides with detergent, but please do not use the water only because that will not completely get rid of the oil on the slide. And second, dehydrate the slides and mount the slides with cytosil or other xylene-based mounting media if you are running brown assay. But if you are running the red assay, please do not do the dehydration step since the red signal is very sensitive to alcohol. Instead, we recommend baking the slides at 60 degrees and make sure the slides are completely dry and then mounting the slides with EcoMount. We highly recommend using EcoMount only here because we find EcoMount is the most compatible with our red signal. Okay, now moving on to fully automated workflow. The fully automated workflow allows automation of all steps from baking to counterstaining. So here is the fully automated workflow, which is divided into 3 main segments. The pre-treatment, probe hybridization, amplification, and signal detection steps are all done online. Here for protocol setup, once again, please refer to the user manual for the recommended conditions. A few points we want to point out here. For setting up the protocol on Discovery Ultra, first thing we want to make sure the correct software is selected in the field highlighted here. And please also check the baking and deparaffinaization and cell conditioning steps because now we are performing everything online. So these 3 steps should be checked. And please pay special attention to the cell conditioning time and the pretreatment time. So for cell conditioning, the last checked box represents the condition that the machine will run that is not in addition to the previous checked time. And also we want to make sure the counterstain time for counterstain 1 is 8 minutes and for counterstain 2 is 4 minutes. And for VENTANA XT, once again, please make sure mRNA DAB Discovery XT software is selected if you are running brown assay, and also double check the baking time and the cell conditioning temperature as well as the protease time. And also we want to select 4 minutes for counterstain 1 and 4 minutes for counterstain 2. So very similar here. If the tissue is new to you, please refer to this table in the manual. And this table is also shown here. So the temperature is all at 97 degrees. And then for cell pellet, it's 16 minutes. And for all other tissues, please start with 24 minutes as the standard condition. So for protease time, we recommend starting with 16 minutes, since this is the most optimum condition we find for most samples, but this can certainly be adjusted if necessary. So once the protocol is set up, before you begin your run, we highly recommend checking a few key points here. First, please check the bulk reagents. Make sure you have enough bulk solution to complete the whole run. And also, please load the reagents and make sure the dispensers are properly primed. Finally, very important, before leaving the instrument unattended, please make sure all reagents and slides are successfully registered. So you have seen these slides in the semi-automated workflow section. The same here. Please wash the slides with detergent, dehydrate, and mounting with appropriate mounting media for your specific assay you are running. So now that we have reviewed a few basics of setting up the assay on the instrument, let's discuss a few guidelines for a successful RNAscope experiment. First, please review the checklist to make sure that all required materials are available before you start the assay. Second, always use control probes and slides. Finally, optimize the RNAscope assay conditions for your specific tissue. Please note here, if you have any questions, please visit our website for more information on getting started. We always recommend customers qualifying your samples using control as the initial step. So first, you need to do a VENTANA assay setup and reagents check on control HeLa slides or 3T3 mouse control slides using both negative and positive control probe. If you get a very strong PPIB staining and a very clean DAB staining, it means that you have passed the VS assay setup and reagents check. Then you are good to move on to sample RNA quality check. So here, during this step, you will stain your target tissue with control probe using either PPIB or DAB. So here's our QC standard. If your sample passed QC, you can then go ahead and run your target probes. If fails, 3 actions you can take. First is verified RNA quality with new samples. And then second, perform assay optimization. So as some of you may have already noted, RNAscope signal is in the form of punctate dots. This allows us to provide you with a semi-quantitative scoring guideline that you may use to score the staining on your samples. So score 1 and 2 depends on the number of dots per cell, have no cluster is involved. Score 3 and 4 depends not only on the number of dots, but also the percentage of dots in clusters. In the event that your RNAscope experiment does not result in expected staining, please follow the optimization guidelines presented in this section to improve your results. So first, let's review the simple 3 steps for assay optimization. When running the VS RNAscope assay on your samples, we always recommend beginning with standard pretreatment conditions first, using control probes on your sample. Based on the signal intensity and background with control probes and tissue morphology, you will then decide whether or not the pretreatment optimization is required. For example, if you observe high background, it suggests that your sample is underfixed. Or if you observe no or weak signal, it suggests that your sample is overfixed. Any of these observations means pretreatment optimization is required. So the first step to take is we recommend adjust offline target retrieval time if you are running a semi-automated assay. Or if you are running a fully automated assay, please adjust the cell conditioning parameter on the instrument. And this is done by 8-minute increments. After this step, you can also adjust the pretreatment 3 parameters on the instrument. But here, please note that we recommend only changing one parameter at a time, since changing both the cell conditioning time and the pretreatment time at the same time may have a significant impact on morphology and signal. So the conditions that you optimize on your samples with control probes can be used on the target probes. So please note, optimization is dependent on the sample only and does not vary with probes. So now let's review some troubleshooting examples and the results after performing optimization on the samples. So the first example we have here, we have run the VS assay on FFPE samples fixed for 12 hours, which is considered as under-fixed samples, using DAT-B negative control probes using 15 minutes offline boiling. So what we have observed is destroyed morphology and high nuclear background, as well as weak hemotoxin staining. So if you observe similar results, our recommendation is to decrease the offline boiling time. So here we have reduced from 15 minutes to 8 minutes. And then so here we have observed intact morphology, and then also the background noise has been removed. So another example here, same again, we are using FFPE samples fixed for only 12 hours. So here we are running the PPIB positive control probe, same using 15 minutes offline boiling. Again, we are seeing destroyed morphology and weak hemotoxin staining. So if we reduce the offline boiling time to 8 minutes, we start to see a much better morphology and a higher resolution of the signal. So this is an example of the fully automated assay results. So here the sample is FFPE fixed for 72 hours, which is considered as over-fixed sample. So here we have stained with PPIB positive control probe for a standard 8 minutes online boiling. So here we have observed low signal intensity and very dense compact nuclei. So we increased the online boiling to 24 minutes, and then we start to see a much stronger signal and a much better signal resolution. Now is another example here for FFPE fixed samples for only 12 hours. So we are seeing very high nuclear background in the DAT-B negative control staining. So here we reduce both the offline boiling time and also the protease time. So now we were able to remove the nuclear background we were seeing before. And now we're seeing an even better tissue morphology. So now moving on to frequently asked questions. So the first question is automated RNAscope 2.5 VS assay compatible with different tissues? So the answer is currently we only support FFPE samples as well as cell pellets on the VENTANA platform. Second, do I need to use RNase-free water to make my buffers? So the answer is no. Molecular biology grade distilled water is sufficient for making buffers. And third, how often do we need to do the instrument maintenance and decontamination? So the answer is please follow the regular maintenance schedule that is recommended by VENTANA for your instruments. To summarize this presentation, we have discussed the key technology features of RNAscope technology and the compatible VENTANA systems and how to set up the assay as well as troubleshooting tips. So finally, I would also like to bring your attention to additional resources available to you during the course of running the RNAscope assay. Please use the support tab on the ACD website to access all the resources that are available on our website. And lastly, we are always happy to assist you with any questions or concerns you may have with regards to your RNAscope assay. Please reach out to us either by calling us or email us or visiting our website. So now we are very happy to take any questions you may have regarding RNAscope 2.5 VS assay. Thank you very much for taking the time to attend today's webinar. Your comments and suggestions to improve this content would be highly appreciated. We do hope you find this information useful and wish you success in your research. Thank you.
Troubleshooting that we've done at MERCK Palo Alto, but just providing an update in terms of what seems to be working well for them. Basically, we heard from the customer that they were seeing weak RNAscope staining on their VENTANA instrument. When we hear weak staining, I mean, it can mean a lot of different things, and so obviously the first thing that we checked is the ribowash bulk. And we confirmed that they have a good lot, so they should be getting good staining, and that shouldn't be impacting their signal. We also considered whether there might be a red chromogen issue, but they're using optimized software for the red chromogen, so this shouldn't really be a factor. So their concern was actually maybe their instrument isn't performing consistently. And when we went there, we ran some control tissues, including our HeLa cell pellet, some tissue from ACD, a human colon cancer sample, which they've been running colitis, so something similar. And both of those stained well. They also told us that actually when they use tonsil, that works consistently well. So they're really just having trouble with other tissues, which led us to think maybe tissue optimization is required for MERCK. So we did ask them just in terms of how they prepare and store their tissue. They did report to us that all of their tissue is fixed in-house, very controlled. They use 10% NVF for 24 hours at room temp, exactly like we recommend. They store their tissue, in terms of how they store their tissue, all of their blocks are stored at room temperature, and it's not really feasible for them to store at 4 degrees, so we did have some concern about RNA quality. They did say that every time they do their staining, they use freshly cut FFPE slides. Okay, so just a breakdown of the tissues that they shared with us at the very beginning. As I mentioned, their human tonsil, it stains well with both manual and VENTANA assays. They had a colitis sample, and this they knew stains well with a manual assay, but they weren't getting staining up to their standards with the VENTANA. So here we just tried a standard optimization of extending the online target retrieval time, and got improved staining, but it still wasn't to the level of the manual assay.
Can you share your screen, please?
Sorry?
Do you show pictures or do you share your screen?
Yeah, unfortunately I don't have them scanned. They're the customer's slides.
Okay, sorry.
This was done at their site. But I will have some pictures of their spleen. So the spleen was actually the most problematic of the tissues that we tried when we initially went to Merck Palo Alto. So they had experience staining this tissue with the manual assay, and it stained well for them. But they got no staining with the VENTANA standard condition. So because it's a tissue-specific issue, we looked at parameters for optimizing pretreatment. So what can we do? Either extending the online target retrieval, just like we had done for their colitis sample, increasing the time online, or trying the manual offline target retrieval. So this is where they were. So this is 2.5 manual standard. So this is what they're seeing with the manual assay. And then 2.5 standard with VENTANA. And so with the standard conditions, they're basically getting no signal. If we tried optimization of target retrieval step, either doing offline, and we tried doing either offline boil or extended target retrieval. So we got a little bit further by doing the offline boil. We were able to see expression in their highest expressing cells within the spleen, but nowhere near the level they were seeing with the manual assay. We weren't actually able to bring up much signal using the extended online retrieval in this tissue, but we did see an increase in the nuclear haze background. Okay, so of the target retrieval optimizations, it seemed like the offline target retrieval, doing a semi-automated assay, is the best option for them. So I know we've gotten some questions about, on VENTANA, how does online and offline target retrieval compare? And so we did also some analysis in-house, just with in-house tissues, just to get a sort of a survey of, is one preferable, or is offline or online preferable? So we did this using a TMA, so it has a variety of tissues on it, one of each of these types of tissues, and then just comparing directly online and offline target retrieval. So for some tissues, these ones that are highlighted with the pink box here, the online target retrieval was actually preferable, and then in other tissues, the offline target retrieval was preferable. So it really does seem to be a tissue-specific thing. While the VENTANA and manual target retrievals are based off of the same formulation, there is the complicating factor of this formulation is diluted with VENTANA's bulk buffers, which changes the chemistry a little bit. Okay, so coming back to Merck, a second thing that we can look at in terms of optimizing pretreatment, and we looked at pretty in-depth for Merck, is protease treatment. So the standard protease treatment is 16 minutes at 37 degrees, but we can also adjust protease activity either by lengthening or shortening the time, by adjusting the temperature to get a more optimized activity out of the enzyme, or by using an increased protease concentration. So the first thing we did here, the easiest thing, is just to extend the time. So that's what's shown in this last column here. And you can see that by combining both the offline boil and an extended time for the protease, we were able to get a lot further in terms of seeing signal in their tissues. Okay, so this is basically, the customer had provided us with a small set of samples, and this is what we had done in-house. So we went back to them and said, we can push it just a little bit further, maybe if we can try adjusting the temperature of the protease or increasing the protease concentration. So just a little bit about why we wanted to do this.
Sorry, can I interrupt again, Emerald?
Yeah.
We're not seeing anything in the WebEx. Are you guys sharing?
Oh.
It was just, yeah, you were presenting.
I thought I was here. Let me.
You're presenting, but I don't know if you're sharing.
I'm not sharing the screen. Oh gosh.
Perfect.
There it is.
There it is.
Much better.
I'm sorry.
I was like, I think I sent it. Can you go back?
Go back
to the target retreat, the different conditions.
Yeah.
Perfect.
Sorry, I should have known, because then it would have popped up for me too. Okay. I was just looking, it's like, wow, and nobody said it.
Okay. So here in this most left column, this is staining that we did here at ACD with their tissue. So this is using the manual standard conditions on their spleen, DAT-B and PCIB. So what they're seeing is basically nothing with the VENTANA standard conditions. If we do offline boil, there's some expression in we can see some expression in the highest expressing cells, but it's just not to the level that they're seeing with the manual assay. And then if we try it, not offline boil, but actually just extending the online target retrieval, we didn't really pull up much signal, but we did pull up a lot of background in this tissue. So tonsil and lymphoid tissues in general are one of the most susceptible to the VENTANA nuclear haze background. And then trying this extended offline boil plus extended protease, we were able to get a lot more signal.
So it's both.
Yeah, it's a combination. Yeah. And I did try extending protease alone with the standard online pretreatment, but that didn't really get us anywhere.
Okay.
Yeah. Okay. All right. So took this back to the customer and asked, do you want us to push it a little bit further? We had some ideas, including adjusting the temperature and increasing the protease concentration. So in terms of increasing the temperature, we currently incubate the protease step at 37 degrees. And we know from the vendor that the activity of the protease is actually relatively low at 37 and can be cooked up higher by increasing the temperature all the way up to 60 degrees. So I've done some studies on this in-house. This is just using the 2.5 Brown assay on VENTANA. I'm doing a temperature titration, and basically we see the most optimal results running at about 50 degrees Celsius. Yeah?
If 50 is the best, why do we use 37? Can we use 50 for all?
Yeah, so
as a standard?
Yeah, so 37 has been working well for most tissues. Protease is kind of tricky because if you under protease treat, you're going to get less signal, and if you over protease treat on some tissues, you'll get less signal. So we don't want to push all tissues to the extreme because we don't want to over protease some tissues.
So this is purely then for just the tougher tissues.
Yeah.
Is this the same protease in the manual assay as in the VENTANA?
It's not the same with the manual and the VENTANA. However, it's the same between LICA and VENTANA. And in LICA, it's not getting diluted on the slide, whereas on VENTANA, it does.
So, but when we usually in the manual assay, we say we have a broad range protease buffer. Is that true, or it's only one protease inside?
So currently, we only have one protease available for VENTANA users. But I'm going to talk a little bit about maybe trying one of our others. Sorry?
I mean the composition of the buffer. Is it one or is it several of them?
I don't understand.
Is it one protease or several different proteases?
She's talking about the enzyme.
Yeah, it's one protease.
Yeah.
It's one enzyme.
And what is the difference between manual and VENTANA then?
The concentration of the protease.
Okay. Thank you. What is this next? You said you mentioned alkylase. Can you go one slide before? Proteinase 26. Okay. Okay. Thank you.
Okay.
Don't share that information.
Yeah.
With others, just to be clear. Don't share what protease we
Okay. So we also looked at whether there might be benefits to some tissues by increasing the protease concentration. So with manual, for some customers, they'll use a different buffer for getting better results on their tissue. So we have other protease bulks available in-house. So one of those is PRETREAT 4, which has more than double the activity of the standard VENTANA protease. So again, this is done on our TMA in-house, just with our in-house samples, just to get an idea of how different tissues how a variety of tissues would respond. So in most of the tissues, there's either it goes too far or there's not really a perceivable benefit. But for one of the tissues in the TMA, there was actually, it did help bring up the signal. This gave us some hope that in some tissues, going stronger with the protease can help bring up more signal. So we wanted to try this with MERCK. So we talked to them, and they agreed in terms of trying to push the protease a little bit further. So they gave us more tissues, and we tried both increasing the temperature of the protease incubation. We tried using a stronger formulation, and they also asked us to try day-to-day reproducibility, just so that they would feel more comfortable with whatever we're recommending to them.
That's the highlight too, though, right? In terms of, not highlight, but one of the major, how do you call this, issue that they had is just the reproducibility, the day-to-day reproducibility.
So they did mention having issues with reproducibility, but after talking with them a lot, my takeaway was they were having most of their issues with seeing the same results on a certain tissue between manual and VENTANA.
Okay.
So for some, they did see that like for example, the tonsil, it looks very similar, but then they were running other tissues where it wasn't looking similar. So this is where going in and actually talking to the customer kind of helped understand a little bit more what they were actually having issues with. Because the first report was just, yeah, weak and variable staining. And what that turned out to mean is they don't always get reliable results on their tissues. Okay. So the spleen that they provided to us is a pretty large tissue, so it selected 5 equally distributed representative points within their tissue to just sort of show how it's affecting their tissue overall. And then looking at either extending the protease time using both extended and heated protease treatment, or using the PRETREAT 4, which has this higher activity. And then they asked for 3 runs at ACD and 2 runs at their site. So this is basically a summary from the results that we have here. So on the left is what we see with the manual staining, and then this right is 3 days using their tissues, and this is so running PPID with the offline boil and extended protease only, the extended heated protease, and then a negative and positive ADAPI and a PPIB for using PRETREAT 4. So overall, it looks to us that PRETREAT 4 is maybe pushing it just a little bit further, getting it towards the level of being more comparable with the manual assay for them. So this is position 1.
What is the position?
Oh, this is to tie back to this map here. So this is just looking at different points in their tissue because I wanted to extract at a level where they can see the dots, but also see give an overall impression of how it's affecting their tissue. Yeah.
Is the customer okay with the red haze? The
right.
Because we have seen that on the DAFI, right?
Yeah. When you switch to PRETREAT 4, it's definitely you see a lot of red haze.
We did see it a little bit, and we talked about this with the customer. The level of haze that they're seeing falls. The signal that they're seeing is much stronger than the haze of the background. So because they could distinguish the signal, they were okay with it. And we did talk about the nuclear haze. So we did see the nuclear haze come up with a stronger pretreatment. And we talked to them about it. They seemed to be okay with the signal level being stronger.
Is it because the stronger the protease you use and then the higher endogenous AP activity you expose? So you see higher haze, you see more haze?
So we've done a lot of investigation into this VENTANA nuclear haze. And we think it has we've narrowed it down to the amplifiers, amplifier trapping. And so we're working on ways to mitigate this. But yeah, we don't think it has anything to do with the endogenous AP activity. We see it actually worse in the brown assay than we see it in the red assay. But most tissues don't have this haze. Only a small percentage of the lymphoid tissues that we've seen have had this haze issue. Yeah. Okay. So this is just a couple more positions, just showing basically using our current protease and extending the time and temperature. We got most of the way there. However, it does seem to still be a little bit on the edge where they're getting a little bit variable results. If we push it all the way to pretreat 4, things look a bit more consistent and more close to what they were seeing with the manual assay. This is another position.
That looks great.
Another position. Okay, so just this is just kind of a summary of a couple of positions and where they've started in terms of their manual versus VENTANA staining and where they are now using the pretreat 4.
Even better than the
Yeah, that is awesome.
Okay. So using this pretreat 4, we were able to get stronger signal and better day-to-day reproducibility on their spleen sample. And showing them this data, they were encouraged, and we were able to bring the optimized spleen protocols to their lab, and we're offering to provide pretreat 4 as a custom reagent to them. So far, they've been really happy with the signal strength that they're getting now compared to what they would expect based on the manual assay. And taking it to their site again, they tried on many other of their in-house tissues, and it seems to be working well for them pretty much across the board in terms of 10 tissue types that they've tested. So I just want to talk a little bit about protease optimization. So I think I mentioned a little bit about this before, but protease is a little bit tricky. If the protease is too weak, you won't get very good signal, and if the protease is too strong, you won't get good signal. So most of tissues fall where the standard VENTANA protease is giving them good results. But there's some tissues, like the MERCK spleen, and it appears maybe other MERCK tissues as well, where it actually needs to be pushed further. So pretreat 4 is not necessarily a one-size-fits-all solution, but for tissues where they're having trouble getting signal, it might be a good thing to try. Okay, so just some notes about using pretreat 4. We're making pretreat 4 available for a custom reagent for Merck and wanted some feedback in terms of whether anybody thinks that other customers may also benefit.
Already. And Lisa at Takeda, they already use it.
Takeda.
Yeah.
Okay.
Yeah.
Okay.
I think they tried that.
How much pretreat 4 is required? Sorry, I may have missed that. Like, what's the volume?
So it's used in place of standard protease, so we would be providing them the same volume as they get in their kit.
I think I don't know the volume off the top of my head.
Yeah, 14 mils.
Yeah, it's like 14. Yeah, 14 mils. 14 mils. Okay.
Yeah.
1/4. Yeah, 1/4.
Yeah. And one big caveat for this is that because of the way the VENTANA software operates, only one protease can be used on the instrument at one time. So they would need to make a choice. Do I want to run standard 2.5 VENTANA protease, or do I want to use pretreat 4? It's going to need to go in the same dispenser as the standard protease would go. So labeling is important so that it doesn't get mixed up with the standard protease. And then in case they are wanting to run also some samples that might be more sensitive to protease, one thing that they can do if they do decide to use pretreat 4 is cut down the time of the pretreat 4 protease incubation from the standard 16 minutes at 37 degrees down to 8 minutes of 37 degrees. This will effectively have the protease activity and bring it closer to the standard 2.5 protease.
So was that what was done for the MERCK spleen?
So for the MERCK spleen, we used the standard conditions, but when we took it to their instrument and we could only run one protease, I also wanted to run, for example, a HeLa cell pellet control.
Oh, and so that's
so if we run HeLa with pretreat 4, the tissue gets totally destroyed. So what I did is just cut down the time, and that worked out really well. Their signal looked good still. The cells were intact. with a normal-looking morphology. So that was encouraging.
Is for in the regular kit. Just tell them for the standard condition use 8 million.
Well, we don't.
No, so we have so that we can make money. Okay.
Well, and also, I mean, I think a fair number of customers are happy with what they have now.
True, very true.
So I think this is more of a specialized case solution.
So this will work for Jen. You had that question yesterday with a customer, right, that had an issue with one of the their tissue.
Sorry, guys, I was muted. Emerald, can you remind me where you guys ended up with on the boiling with this pre-treat for? I know you told us, but.
Yeah, so they're still doing offline boiling, and then they're also using an optimized protease.
Okay.
So what do you think? I mean, I have a customer who's really wants to try to get things working online. Do you think that this would be something that we could offer him, or do you think without the offline boiling, it's not really worth trying?
Oh, so I think in the case of the MERCK samples, they really did require both. Yeah. However, that doesn't mean that every tissue is going to require both.
Sure.
So we had a hard time with the MERCK samples in particular getting any signal at all using the online target retrieval, but that's a really unusual, atypical case. So I think it's definitely worth trying even if they want to stay with offline target retrieval, especially because if they want to stay with online target retrieval, then the options for pre-treatment optimization are a little bit more restricted. This might give a little bit more options of what to try for them.
All right, yeah, I can talk to him about it. And so if that comes to an interest, is this something we're just giving as a support item, or are we actually going to sell this?
Sell it, right? Yeah.
Sell it? Okay.
Yeah, sell it.
So then I should get Shab involved and figure out how to code it?
Yeah, so you code it as a custom reagent, Jen.
Okay.
But I think, you know, I'm thinking about your friends at Novartis. I mean, let's I assume that's what you were thinking of Novartis. I'm
actually thinking about Pfizer. There's a customer at Pfizer who's messing around with it at Andover. You know, Novartis, I will certainly mention it to them, but they don't have the appetite for all this optimization work.
Right.
Unless we can guarantee it's going to work, you know, they might try it once, and then if it doesn't work, they're done.
Yeah, exactly. So use your discretion. Obviously, we definitely want to charge for this because it is larger. The biggest reason is that it's bulk, right? And we're just not set up. Right now, it's bulk before we actually make this into a real product for this particular volume. But it's definitely on our radar since, as Connie mentioned, we've got our friends at Takeda on the Leica side are also using this for their specific use, which is very different from here. And we'll just keep an eye on it.
Sorry, I got Friday brain already. So is this pre-treat 4? Is it the same pre-treat 4 that's in the manual kits, or is this a unique formulation? Okay, so if we just order pre-treat 4, we'll get the right thing as a customer reagent. That was my main.
Correct. It's the protease 4, just higher volume. And so, and if you don't mind, just CC me so that I can see so I can give Dan a heads up as well.
Okay.
Hey, Jen.
I mean, I'm going to present it as, hey, this is something we were working on with another customer. It showed, you know, promise. We actually solved the problem with it, but in a combination with offline boil. So, would you be interested in trying it? And then they could say yes or no.
Okay.
Yeah.
Go ahead, Tess. You had something?
I would like it here. Jen, I would love to bring it in-house, but right now we're pretty tight with the Leica instruments. Right. However, if it is that this can wait till October, that's fine. But if we need revenue, then I'll put it on your end to decide or what the customer says. But for Novartis, I don't know if they have their instrument replaced. I heard it through Wendy that they could possibly get 2 new instruments. I'm not sure, but if not, we can try that here with Emerald to run their samples and try this optimization for them. If they're interested.
I can offer it. I'm certainly, I mean, Joyce isn't the type that, she'll either say yes or no pretty much. She'll probably say, sure, we can throw you a few slides. That's no skin off our nose. So, I'll approach her, but I'm not in a super rush because they're pretty stable. I don't want to mess with it.
Okay, sounds good. Thanks, Jan.
As far as Pfizer's concerned, it's Zach. I forget his last name, but he's kind of he's a tinkerer, so he'll actually want to do it himself because he likes to look up that stuff. So, which is great for us. So.
Yeah, definitely.
Okay. So I also wanted to just kind of give a little bit of an overview in terms of recommendations from the R&D side and in terms of VENTANA troubleshooting. So if a customer is complaining about small dot size, for example, I think we all know for VENTANA the AMP5 time is a major way to address this. But if the dot size changes dramatically overnight and the customer reports just a really severe loss of signal, I think the go-to thing to start out with is to check the RiboWash number, lot number. However, if they're seeing low or no signal, pre-treatment optimization could be beneficial. In terms of target retrieval, things that can be done include extending the online target retrieval time, trying offline target retrieval, and then protease. I think we've found through this MERCK troubleshooting that actually we can get pretty far using protease optimization, either extending the time, combining extended time with increased temperature, or even going as far as to use the PRETREAT 4 custom reagent. So those are my recommendations.
Yeah.
And Emerald, I've never paid attention at the protease step, but I'm assuming the customer can adjust the temperature on the fly in that protocol?
Yeah, they can.
Okay.
And the time can be increased up to 32 minutes.
Okay.
Okay. So I think that's all for me.
All right.
Thanks, Emerald.
Nice. Thanks, Emerald. It's a lot of work.
Yeah, and it's great information, so you know I'm going to ask for the deck so that I can review it later. I will digest this.
That's perfect. We can give that to you. And we've also recorded this. So I think Emerald, can you just, for the sake of being careful here, in terms of just put confidential on one of the slides?
Sure.
That'll be good. And then
it's mostly for me to study before I go in and start trying to work with a customer.
Yeah. But we're uploading this on the knowledge database, and everyone has access to it. I just want to make sure that we're covered here. Okay, you'll send this out once you have that. You guys have any questions or any anything to share? So I'm going to start recording this session, and we'll have a support area somewhere. I'm not sure where, and any discussion that we have or updates, we'll have this on the support server somewhere. I'll talk to Dan about it. I know the last meeting that was also recorded, we had the show pad and also update from Vinay. I think Vinay, you updated us on the LS multiplex 2.5, so that'll also be uploaded on that server somewhere. But so I'll do this on the consecutive meetings that we'll have. Okay.
Okay. And also, speaking of LS Multiplex, one last bit is we're creating the tech notes for LS Multiplex plus IHC. And I think I may have passed it around. I can't remember right now because I have so many documents floating through. Oh, sorry. Right now it's just going through BQ first, and then I'll pass it around to folks on the phone, and so you can just get familiar with the tech note.
Okay.
Thank you.
The last thing to share outside of our technical updates here is that we have Chuck, our Bio-Techne CEO, and then our VP for Bio-Techne HR, Stuart. I can't never. And then a guy named Jack, and I don't know what his position is. So they were here yesterday, and they're here as well. Well, we have a meeting at 10, right? The other thing too, though, is that several of us have visited Protein Simple. They had a barbecue yesterday, so some of us did visit the area. And it was a huge, nice Scandinavian type architecture. I'm saying that based on what I think it is, but I think there is a lot of pretty interesting.
Looks like IKEA. It looked like IKEA, you're right.
Thank you. Yeah, it was pretty cool. They were pretty excited as well for us. And so I think in small groups, we did share what we have from our end and then ask questions as well as to what impact. If there was any, I don't know if they would have shared that in terms of, Bio-Techne purchasing them and all. But the cool thing, I thought the biggest, two things, that they have an awesome bar, right? I think some of you saw that. And then there's an area for a meeting, a group meeting or company meeting, and it was pretty spacious. And I don't know what you call that screen. What type of screen is that? It's humongous. It's like a movie center. It's amazing. And then the last thing outside of physical look of everything else, I think the last thing that we had also gotten information on, but I don't think this is reliable because the person that took us on a tour was a generalist, an HR generalist, but I wasn't too sure in terms of the structure of the operations to the commercial team, if there was any commercial, or the PM and marketing, whether that was separate or or a similar marketing/PM, and then from the operations and support, what's in that operations team. So it's totally different than what we have here. They had manufacturing. They had at least 20 people in manufacturing. They have both consumables as well as the instrumentation. So when you guys have a chance, when you're here, I think they're open to us visiting them. So, all right. We have to jump to that meeting, you guys, in 4 minutes. But have a great weekend, and we'll see you on Monday somehow, somewhere.